Hello, this is the final installment of teas, tinctures, and tablets. And hasn't it been a long saga? But it's really worthwhile understanding these fundamental issues. So, here in part five, we are going to finish off the all important topic of standardized extracts, understanding standardized extracts, part two. So, definition and the pros and cons of standardized extracts we covered in part one. And now, marker versus active compounds, this interesting concept of phytoequivalence, and the basic types of standardized extracts, just how we can understand what an individual standardized extract is of a particular herb. All those three I'll be covering now in this brief presentation. So let's begin with marker compounds. What are they? Well, they're characteristic phytochemicals given in a plant that are chosen to represent a quality standard, both for the plant, the original starting material, the raw material, and the extract that's made for it. And often they're what the extract is fixed or standardized to. It doesn't mean necessarily, and in most cases not, at the exclusion of other important phytochemicals in the herb. It's just that you're fixing to certain marker compounds for that consistency. Now, the drawback is they're not necessarily active compounds. So you can still get variable activity when you fix to marker compounds if the true active compounds are elsewhere in the plant and in the other phytochemicals, and they're not being fixed to a consistent level. So for this very reason, they must be chosen quite well to serve a useful function in terms of quality and applied overall throughout the manufacturing process in terms of identification, ensuring appropriate drying, handling, and extraction of the herb. What happens when you've chosen some marker compounds is you generally set the raw material, the herbal material, the natural plant material, to contain a minimum level. And if it doesn't contain that minimum level, you reject it. So specifying on a marker compound can lead to consistent extracts if a consistent manufacturing process is then applied, and hopefully that will translate to consistent therapeutic activity. But as I said, it depends on how well the marker compound is chosen or compounds are chosen. So active compounds, by contrast, they are the phytochemicals that are actually important for contributing a therapeutic effect of a herb or indeed its extract. And we often don't really exactly know what they are. But wherever possible, marker compounds should have some activity. They should also be active compounds. Now, what's missing from that statement is the. Wherever possible, I'm not saying wherever possible marker compounds should be the active compounds, because it's far more complex than that, but they should be among the phytochemicals that we think contribute to the therapeutic activity of the herb. And it's always worth keeping in mind, as I often emphasize, that herbs are chemically complex, and the active component is always the whole herbal extract itself, or at least the extract that you're giving to patients. So this begs the question, well, how in fact do we choose marker compounds? Well, there are many sources of information we can draw on. The first is industry or pharmacopoeial standards. So sometimes, like in the European Pharmacopoeia or the United States Pharmacopoeia, the USP, it specifies that a certain herb must contain certain levels of marker compounds, and indeed quite often it will also specify that an extract from that herb given at a specified dose must contain certain levels of marker compounds. We can do original phytochemical research and identify what is actually in that herb that's likely to have therapeutic activity. And that can be backed up then by the pharmacological and clinical literature that suggests, yes, indeed, these phytochemicals do have activity. But that must always be tempered by a knowledge of how the herb is used therapeutically and therefore what's likely to be active in that particular clinical context. Pharmacokinetic considerations are also very important. And I think this is well illustrated in the case of echinacea root, because generally when you consume echinacea root, the only compounds that can be detected in your bloodstream that were originally in the plant are these alkyl amide compounds. So clearly they must be important for activity because they're the only compounds that actually reach remote tissues in the body. So summing up, in practice, it's best to select marker compounds that most reflect therapeutic activity from a well-considered, thought-through position. And often it's best to choose more than one for a given herb. And I've used that here in the image illustration because you see these are phytochemical marker compounds that are found in the herb ginkgo biloba. And you see you have the ginkgolides, bilobalides, and the flavonoids. And they are all now used together as marker compounds to define ginkgo extract of good therapeutic quality. I'm now going to talk briefly about this concept of phytoequivalence because it is actually, while you might on the surface of it think it's rather abstract, it is actually quite important, as I hope to illustrate. So phytoequivalence is where two herbal extracts from different manufacturers can be expected to have the same therapeutic activity. Usually the manufacturer or the clinician who's using the second extract or the manufacturer of the second extract wishes to draw on the clinical efficacy data for the first. So I personally encounter this issue often. So I might read a clinical study that shows a herb is really good for something that we didn't expect it was good for. And I immediately look at, well, what was used? What was the dose? And can I replicate the type of extract that was used, given that it might have been a study, for example, in Iran, and I can't get that particular product? How can I replicate and ensure that I will get the same efficacy? So you have to invoke this concept of phytoequivalence. Well, yes, maybe the extract I'm using from a local supplier is phytoequivalent to what was shown to be effective in that study. It's a complex issue when applied in practice. At the very least, the second comparative extract should closely demonstrate the same phytochemical spectrum as the original extract. Not just the marker compounds, but hopefully the full phytochemical spectrum that you can see, for example, on a high-performance liquid chromatography trace. So in other words, not just marker compounds should be the same, all other measurable compounds in the extract should, as far as we can determine, should be present at similar levels. And ideally, and this is often applied by regulatory authorities, especially, for example, in Europe, the levels of the marker compounds or the derivatives achieved in the bloodstream of humans after oral doses, in other words, a pharmacokinetic study, should also yield similar results between the two extracts. And that's certainly applied, as I said, when a company in Europe, for example, wants to make the same therapeutic claim to consumers or practitioners based on the study of another extract. So here's an example. Here's a willow bark extract, and you can see we're comparing the red one, which is the one that was used in the clinical trial, with the green one, which is, well, will this extract produce the same kind of anti-inflammatory effects that were demonstrated in the clinical trial of the red one? And as you can see, the green trace, or it's not totally exactly the same in overall spectrum, the HPLC spectrum, as we call it, high performance liquid chromatography spectrum. Overall, it's quite a good match. So we can say that it's very likely that the green extract is phytoequivalent to the red one. Now, I'd like to finish off by giving you something that I think is very important, and that's a basic understanding of how standardized extracts are made, and we can classify them. And this is based on my thinking around the topic. It's not necessarily, documented in articles, but it's what I've seen from my many, many decades of extensive experience in herbal manufacturing. And there are three basic types of standardized extracts. They are the galenical type extracts. So I refer you back to earlier presentations in this theme. If the term galenical extract doesn't mean anything to you, but basically it's a standard alcohol water extract, a tincture or a liquid extract of a herb. So dried galenical type extracts form, in fact, the majority of standardized extracts that are available in the marketplace. Then we have the examples, which are fewer, but some important ones, of highly concentrated extracts. And then finally, we have the examples of selective phytochemical extracts, where they're also highly concentrated and they select out just a particular phytochemical fraction from the herb. So they're deliberately made not as whole herbal extracts. So I mentioned in the previous presentation about the pros and cons of standardized extracts and the controversy and the extreme viewpoints, where some see they're the future of herbal medicine and others see them as literally the work of the devil. But I think we see that reflected, and those issues reflected across these three types of standardized extracts. Because to me, the galenical type extracts, no purist should have any objection to those at all. They're simply dried tinctures or liquid extracts made to a consistent standard of marker compounds, which is a good thing to do. On the other hand, the selective phytochemical extracts might certainly be seen by, call them purists, as I have, that they don't represent true herbalism. But it's fascinating because I was having such a debate a decade or so ago with a well-known US herbalist about this issue. And she was saying, I have a real problem with these selective phytochemical extracts. They're not true herbal medicine to me. And I said, hang on. I recall you use essential oils in your practice quite a bit. Oh, yes, she said, I love essential oils. They're so important for my practice and they deliver such great therapeutic activity. Well, I put to her, and she agreed to her surprise when I put it this way, essential oils are in fact selective phytochemical extracts. You're isolating one fraction from that essential oil. Okay, they're made by steam distillation mainly, but you can do a hexane extract of a herb and come up with something that's very close to a steam distilled essential oil. So they are selective phytochemical extracts. And that gave her pause, at least, to think, well, yes, well, maybe they're not such a bad thing after all if they're used appropriately and they have good clinical evidence and they're safe to use. So a bit more detail then about these three types of standardized extracts. The galenical types, as I said, they're just dried galenical extracts fixed to a level of marker compounds, and they're not selectively concentrated and therefore usually have concentration or extraction ratios less than 10 to 1, and they're typically in the range of 4 to 6 to 1. And as such, they are definitely, without doubt, whole herbal extracts. And they dominate. And there are many examples: St. John's wort, horse chestnut, gotu kola, many, many others are that type. Then we have the highly concentrated extracts, and you can see from my image there, ginkgo biloba is one of those. And here the extract is usually much greater than 10 to 1. And they're extracted often with solvents, not necessarily, but solvents different to ethanol water mixtures. And they might, unlike the galenical type standardized extracts for which it's ethanol and water, they might use acetone, liquid carbon dioxide, hexane if it's more essential oil-like, for example, or more fatty-like. And additionally, they will employ multiple solvent extraction steps involving ethanol water mixtures or the other solvents such as those listed above, or they will use standard chemical isolation techniques such as chromatography columns, ion exchange resins, precipitations, and so on to achieve that high concentration ratio. So examples here include ginkgo biloba and the better forms of Tribulus terrestris, that's made from the Eastern European leaf that's used for its hormonal effects. So ginkgo is typically 50 to 1 and Tribulus 20 to 30 to 1. But personally, I, of course, have no hesitation using these types of extracts in my practice, provided they come from a reliable manufacturer and they're tested for solvent residues, which should be not there. And then we have the selective phytochemical extracts, which do, as I mentioned, include essential oils. And here, as mentioned, one particular phytochemical group is selectively removed from the herb. And this can involve any of the processes I mentioned earlier that are used to make the highly concentrated extracts. And as a result, often, but not always, but typically they're highly concentrated. And I've mentioned about essential oils. And examples here include the grapeseed extract, the saw palmetto liposterolic extract, and the silymarin extract from milk thistle. Now, it's really interesting if we look at the saw palmetto liposterolic extract. That's basically a fatty extract from the saw palmetto berry. And it can be made with either hexane, 80% ethanol water, or liquid carbon dioxide. And you end up with extracts that are largely identical, although the manufacturers of the hexane extracts claim they have the best clinical evidence, which is true, but that might be just because of the trials they've done. But they're more or less identical. So it's interesting that 80% ethanol water is more galenical-like, and yet with the more modern solvents like hexane and liquid carbon dioxide, you can achieve a similar thing. So those three types of versions of saw palmetto liposterolic extract are available on the market. And unlike essential oil, it's not a volatile sort of fatty extract. It's more like a cooking oil in that sense, in terms of its texture and its components. So thank you very much. We have finally finished the topic of teas, tinctures, and tablets. And in my next presentation for Herbal Basics, I'll be looking at some important concepts in herbal pharmacology. I hope you can join me for that.